So our next topic from the chapter Biotechnology, okay, from the unit is RNAi. Here I stands for interference, okay. I stands for interference. So RNAi means RNA interference. Now how disease resistant plant, okay, how disease resistant plant are obtained from RNA interference, that is the another point, another question, okay. How we can obtain disease resistant plant are which plant which are resistant to diseases or any kind of pathogens, okay. How that kind of plant are obtained from this technique, that is RNA interference. Now interference means to stop something, to distract something, okay. But here, now what is RNA interference? RNA is being distracted. How? Okay, we are going to know about all those things when we are going to learn about the steps involved in this RNA interference in order to produce disease resistant plant. Now, so the steps involved first here is mRNA from the pathogen, messenger RNA, mRNA, okay, from the pathogen or the parasite is converted into cDNA. mRNA from the parasite means disease causing organism or pathogen sometimes, okay. mRNA from parasites or pathogen, it is converted into cDNA, okay, cDNA by the process called reverse transcription. You know that transcription is the process in which a DNA is converted into RNA, but here the RNA from the pathogen, it is converted back to cDNA, and that process is called reverse transcription, okay. It is reversely transcripted to cDNA, DNA, okay. Now number 2 is double stranded cDNA. Now you know that DNA is double stranded, okay. So when mRNA is converted to cDNA, cDNA is double stranded. So double stranded cDNA is introduced into host plant using agrobacterium vector, okay. So this cDNA, which is double stranded, okay, it is then introduced into host. Now host here is plant that we want to have a disease resistant property, okay. The main plant, it acts as a host and vector. Always remember, you already know that in biotechnology there is gene of interest involved, okay. Gene of interest is involved, then vector DNA is involved, which together makes recombinant DNA, okay, and the same is introduced into host plant, the targeted plant, okay. So the cDNA, which is double stranded, okay, that DNA, now DNA is nothing but a gene, okay. So that DNA is now introduced into host plant using agrobacterium vector, using bacteria, okay, soil bacteria as a vector. They form, first of all, cDNA along with the vector DNA, that is soil bacterium DNA, they form recombinant DNA, and that is introduced into host plant, all right, okay. So number 3, third step is cDNA introduced into the host, expressed inside the plant. Now this DNA, which is nothing but a gene of interest, okay, this DNA is this one, gene of interest, okay. The cDNA, which is introduced into host plant, this DNA is expressed in the host plant because it has already gone into the host plant cell, okay. So now it will be expressed. So when this cDNA is expressed inside the host cell, they form, now when cDNA is expressed inside the host plant, okay, our target plant, that plant, now when it is expressed, then now this cDNA again forms RNA, because we know that from DNA, RNA is formed through the process called transcription. And when this DNA is expressing inside the host plant, that means DNA is active. So when DNA is active, it forms, first of all, it forms mRNA, and from mRNA it gets converted into protein. Okay, so cDNA will be expressed. Now the mRNA that will be formed, okay, when this cDNA is introduced into host plant, the mRNA which is formed from cDNA will be double stranded. Okay, they form sense and antisense strand. They will be double stranded, number 1, number 2. Okay, these two are not single but double stranded. So the mRNA will be double stranded. Okay, the mRNA which is formed from cDNA will be double stranded, and then sense and antisense, these two strands, okay, they are complementary to each other. That means if in one strand there is uracil, guanine, cytosine, then in the other strand, antisense strand, it will be complementary. Opposite to U will be A, G will be C, C will be G, like that. Only thymine will not be there, because you know that in RNA thymine is not present. Instead of thymine there is uracil. Okay, so it will be complementary. These two strands will be complementary to each other. Okay, now when these two sense and antisense strand of mRNA are complementary to each other, that means they are going to form double stranded RNA. Okay, double stranded RNA. Now double stranded RNA binds to the specific mRNA of parasite. Now it is done. When again some other time, if pathogen, okay, if any pathogen comes in contact with the host plant, okay, later on when any pathogen comes in contact with the host plant, what happens is that this double stranded which is already present in the host plant, double stranded RNA which is already present in the host plant, okay, they binds to the specific mRNA, because when pathogen comes in contact with the host cell, pathogen also has their DNA, okay, then RNA, mRNA, then proteins. Okay, pathogen also has its own DNA. Okay, and when it is living, when it is live, okay, and when it comes in contact with the host plant, their DNA, it's transcribed into mRNA, and after that it is translated into protein, because we know that every living organism goes through these three processes. Okay, but when pathogen further it comes in contact with the host plant which already has this double stranded mRNA, double stranded RNA, we can say RNA also, so double stranded RNA, then it binds to this specific mRNA of the pathogen. When any pathogen comes in contact with the host plant which already has double stranded RNA, this double stranded RNA binds to the mRNA of the pathogen. Okay, now what happens when it binds to the mRNA of pathogen? It interferes with the translation process. When this double stranded RNA binds to the mRNA of pathogen further, then this will interfere with the formation of protein. This will interfere with the process of translation. There will be no translation of this mRNA into protein, because this mRNA is binded to the double stranded RNA which was already present in the host plant. Now when this mRNA of the pathogen, they cannot be converted into protein, what happened? The parasite, this parasite or the pathogen, disease causing organism, they cannot live in the host for long, because mRNA is not translated into protein. Okay, it is interfered by the double stranded RNA. So when this is not translated into protein, the pathogen or the parasite or the disease causing organism, they cannot survive in the host plant for long, they die. Okay. Thus the host plant, okay, the targeted plant is protected from pathogen or parasite. This way it is protected. Okay, and it is all done through biotechnological process, because there is cDNA, formation of cDNA, okay, then recombination DNA, then that DNA is introduced into host plant, which makes sense and antisense mRNA. Okay, when they interfere with the mRNA of pathogen, then only there will be interference. So this is all about RNA interference. Okay, I hope it is clear now. All right, so the next topic now we are going to do is humulin. Okay, next topic that we are going to do is humulin. Humulin is nothing but human insulin. You know that insulin is present in the pancreas. Okay, it is nothing but the protein. Insulin is nothing but a protein that is present in the pancreas of human beings. Okay, humulin. Now, how humulin is produced biotechnologically? That means artificially how humulin protein can be developed or produced. Okay, now let's see the concept. Humulin is an human insulin which is produced by genetically engineered E. coli bacteria. So, humulin, it is artificially produced insulin, human insulin. Okay, artificially produced human insulin, and for that E. coli bacteria is used. For the same reason, E. coli bacteria is used. Artificially produced, that means genetically engineered protein. Okay, biotechnologically it is produced artificially using E. coli bacteria. Now, insulin is secreted as proinsulin consisting of A and B polypeptide. So, insulin, this insulin, it has got A polypeptide. Okay, this is suppose A polypeptide, and suppose this is B polypeptide, and there is another type of polypeptide. Suppose this is A. You can see these things in your book. Okay, diagram is given. Suppose this is B and this is C. Okay, so insulin is secreted as proinsulin. Okay, consisting of, this is A polypeptide. Polypeptide, you know what is polypeptide, with long chain of amino acid. Okay, so consisting of A polypeptide, this one is B polypeptide. Okay, which is connected by C polypeptide. This one is C polypeptide. So A and B is connected. A and B is connected by C polypeptide by disulfide bond, S disulfide bonds. Okay, so insulin is secreted as proinsulin consisting of A and B polypeptide connected by C polypeptide of 33 amino acids. Okay, polypeptide is nothing but the long chain of amino acid which is linked by disulfide bonds. Proinsulin, it gets converted into mature insulin by cleaving C polypeptide which is absent in mature insulin. So, proinsulin is not mature. Okay, this is the diagram of proinsulin when it is first made. Okay, which is not mature. The initial insulin which has got A polypeptide, B polypeptide connected by C polypeptide by disulfide bonds, and in the mature polypeptide this C polypeptide is cleaved. That means it is broken down. Only A and B will be left. A polypeptide and B polypeptide will be left and the C will be free. Suppose this is A and this is B, and C will be cleaved. This polypeptide will be cleaved in the mature insulin. Okay, this is free C polypeptide. Okay, now how it is produced? Let's see the steps. Okay, how it is produced biotechnologically. Now, number one is prepare 2 DNA sequences for A and B polypeptide, because before forming polypeptide, okay, polypeptide is formed, you know that long chain of amino acid it is formed. At first DNA will be converted to mRNA, and from mRNA only then the polypeptide will be formed. So prepare 2 DNA sequence. We need to prepare 2 DNA sequences, okay, for A and B polypeptide. Okay, now construct recombinant vector using plasmid of E. coli. So this DNA, it should be incorporated into vector first in order to make a recombinant DNA. You already know that gene of interest, whatever gene we are using artificially, okay, it first needs to get into the vector DNA, then only it can be transferred to the host. Okay, we cannot directly inject this gene of interest into the host cell, which I have already told you before also. So we need to construct recombinant vector. So here vector is used as plasmid of E. coli bacteria. It is used as a vector, okay, vector, thereby producing recombinant DNA, transgenic E. coli, because this bacteria E. coli, it is now transformed. It has now transformed because this E. coli bacteria, it not only contain its plasmid, okay, this DNA, but it also contain DNA, that is foreign DNA, which is sequenced or which is extracted artificially for A and B polypeptide. Okay, now then transform E. coli with recombinant plasmid vector having foreign insert. So this E. coli is now transformed. Why it is transformed? Because it has not only its own DNA but also the DNA that code for A and B polypeptide. Foreign DNA or gene of interest is also present. It has got recombinant DNA, so it is transformed. Okay, it has got DNA for polypeptide A and polypeptide B as well as its own DNA, plasmid vector, plasmid DNA. Now transformed bacteria grown in commercial scale. Now this E. coli is already transformed. Okay, it is already transformed. Now this E. coli bacteria is grown in a culture. It is grown in a culture. Why it is grown in a culture? So that it can produce multiple number of E. coli bacteria. It can reproduce many E. coli bacteria which contain both its own DNA as well as DNA which code for A and B polypeptides. Okay. Now extract the polypeptide A and B. When this E. coli bacteria is cultured, okay, there will be number of E. coli bacteria, not only one single. Okay, and after it is cultured, after it makes multiple copies of E. coli bacteria, after it reproduces and makes lot of progeny, okay, after that then extraction, there will be extraction. So we need to extract A and B polypeptide, A and B polypeptide, okay, from this culture, from these bacteria we need to extract A and B polypeptide, because this bacteria, okay, these multiple bacteria, they not only have their own plasmid vector, plasmid DNA, but they also have DNA, okay, that is gene of interest which code for A and B polypeptide. So after it is cultured, we need to extract A and B polypeptide and then we need to purify them. Okay, we need to purify them. Why we need to purify them? Because they not only contain A and B polypeptide, but they also contain peptide of their own. okay, peptide of their own. So we need to purify them. So combine them by creating disulfide bonds to form humulin. Now at last we need to combine this A and B polypeptide, okay, with the C polypeptide, okay, by using disulfide bonds, SS, okay, by using disulfide bonds. Now this will be the human insulin which can be used by human beings, okay, insulin which is present in the pancreas. This way, okay, artificial human insulin, or in short form we can say humulin, can be formed in a laboratory, okay, by the process, by biotechnological process, or we can say by genetically engineered processes. Humulin can be formed, or it can be produced in a large scale. Later on it can be used by the patient, okay, who are undergoing diabetes or any other kind of disease related to pancreas, disease related to insulin. So this is all about humulin.